this CRISPR tool, which was the basis for the Nobel Prize in chemistry for this current year. We have with us a very eminent person to explain about this entire work. Welcome, Professor Debojyoti Chakraborty, to this webinar. And you will understand that our audience will be a mixture of people from chemistry, engineering, life sciences, and many other subjects. But everyone is keen to know what exactly is this about. So I am sure that your lecture will provide us a window to this subject. And welcome to everybody. I request our Associate Dean, Professor D.K. Maiti, to introduce the speaker, and we will go to the lecture after that. Thank you.
Thank you, Honorable Vice Chancellor. It's on my behalf and on behalf of HBNI, I welcome again Dr. Debojyoti Chakraborty, who is a senior scientist at CSIR Institute of Genomics and Integrative Biology, New Delhi. He has done his PhD in molecular biology from Max Planck Institute of Molecular Cell Biology and Genetics, Germany. Then he has done the postdoctoral research at Medical Facility of Technical University, Germany. He has a few awards. He is the recipient of INSA Young Scientist Award, CSIR Young Scientist Award, Mark Young Scientist Award, Lady Tata Young Investigator Award. More than that, most importantly, I think many of you know he has developed a product, and our health minister, I think, introduced it. He called it Feluda, a paper strip for coronavirus detection, which is already available in Delhi market. I think this week it was supposed to come to other cities also. So all of us would like to hear Dr. Debojyoti Chakraborty, please.
Thank you. I'll first share my screen. You can tell me if you can see my screen, and then I'll just start. Can you see my presentation?
Yes, we are able to see it.
I'll just make it full screen. Okay, now you can see the full screen, right?
Yes. Yeah, that's right.
Okay. So thank you once again, Dr. Debojyoti Chakraborty, whom I know for a long time, as a kind of mentor, I would say, and to everyone who as a part of this, the HBNI, Dr. Maiti, for inviting me. First of all, I must clarify, I'm not a professor. I'm very far away from being a professor. I'm a very young scientist, still learning the field more than anything else. So that's why it's an added honor that you have invited me. I guess because I work on CRISPR, that is the main reason. But I would talk a little bit about CRISPR technology. In addition, I'll also talk a little bit about what my lab is actually doing. So once again, thank you. I will just have arranged the talk today in a format where I would introduce a little bit about genome editing, because I think there's a significant portion of the audience probably who are also undergrads, I'm told, and graduate students. So maybe something that what the field is about. And then slowly I will go into the work that we have been doing in the lab. So I think it doesn't have to be told anymore that the field of CRISPR was awarded the 2020 Nobel Prize in Chemistry. What has been a kind of matter of debate for a long period of time was that who would be awarded and when it would be awarded. I mean, it had to get a Nobel Prize. That it was almost certain from the day that the big papers and the big discoveries and in the field of genome editing were made. And this is what I'm talking about, 2012, 2013. But what is, and of course, introducing Jennifer Doudna and Emmanuel Charpentier, who did path-breaking work in trying to bring all this into a format which would allow, in a way, to rewrite the code of life. However, what I would want to emphasize here is that we are not here because of the work principally of this two amazing scientists, but the field of CRISPR has got multiple heroes. And just after the Nobel Prize announcement, somebody actually put this up on Twitter. I found it very nice of this to have this series of people who really have contributed immensely to this field. And this field had started in the 1980s, when the first discoveries about CRISPR in a very rudimentary fashion were made, regarding how certain types of curd would have bacteriophage resistance bacteria, and then somebody going into that sequences and finding some completely weird sequences within the bacterial sequence which did not have any origin in the bacterial genome. And then people began to realize that they were not able to explain what these sequences meant, and going deeper and deeper into it. The story of CRISPR, I think you will have a lot of books, lot of articles that have been written about it. It's very interesting. I can almost certainly recommend that you can go through the timeline, how these discoveries were made. It also told us a little bit about how publication and the concept of publication in what is called so-called high impact journals can also lead to pushing a field back by almost 20 years, because the first studies were sent in Science, Nature and Cell, and they did not get accepted. They got into a different journal, much low impact. And then almost for 10, 15 years, nobody really bothered about these foreign sequences in bacteria. So yes, the field of CRISPR is standing on the shoulders of giants, of people who have worked tirelessly for the last 20, 30 years and are still working. And if you look at the third row in this figure, in this Twitter image, there is also an Indian there, Prashant Mali, an IIT Delhi graduate who worked with George Church and actually had one of the first pioneering papers on CRISPR. And one of the reasons why there was a debate about who would get the Nobel Prize was because there seemed to be a lot of people who have applied CRISPR in human genome editing, which also has a huge impact. So I would again bring your notice to the citation here, the Nobel citation, which says for the development of a method for genome editing. Which is a bit different from application of genome editing in a wide variety of different diseases and different contexts, in which I think George Church and Feng Zhang from MIT, Broad MIT, who also happen to have the CRISPR patents. That's another story altogether. In safely, at least in the first phase, the patent right seems to be belonging to Broad MIT. So there are, as you probably are able to understand, that there are so many different aspects of this entire process, whether it is about the person who first noted in the laboratory notebook, which went on to get a patent, versus the person who really showed to the world in the form of a publication. And the CRISPR field has taught us, so many different things, which are beyond just the concept of discovery, but also about the concept of how well you can really write down your findings. I think that's something that is probably a take-home message for a lot of young undergrads and young scientists. So, what is CRISPR? It seems that the bacteria, as in most cases, are smarter than us. Viruses are, of course, smart because they have almost shut down the whole world for the last year. But the combination of a bacteria and virus is deadly. So one of the three heroes of CRISPR are actually the bacteriophage, the virus, and of course the archaea, which also harbor CRISPR systems. So CRISPR is essentially a type of bacterial adaptive immunity. So just like we have an immune system where when we are exposed to a foreign object, we develop a kind of immunity which helps us in fending off this kind of infection later on. I think with the SARS-CoV-2 infection and the concept of vaccines being there on almost every national newspaper and channel, the general public has begun to understand what is immunity and how that works. So it was not known for a long period of time that bacteria have some kind of an immunity as well. And for them, the foreign object here is the virus. And a virus which attacks a bacteria is, of course, called a bacteriophage. And I have taken this slide from one of Jennifer Doudna's presentations, so I just acknowledge that. And this bacteriophage, when it attacks a bacteria, it generally puts its DNA into that bacterial cell, which gets it gets integrated, and then multiple virions are produced, and then the bacteria is destroyed. So that's a normal way by which a virus actually attacks a bacterial cell. But it seems that certain bacteria have evolved a way by which they can somehow tag this DNA a bit, and it kind of puts this DNA into a very, very specific DNA locus, which is called the CRISPR locus in the bacteria. A locus is a segment of the genome in which this small piece of DNA is actually, taken out and introduced. And you can almost feel like a librarian here, where a new book comes into the library, and you, for safe cataloging, you put in a label there, so that you can then call that back and say that, okay, this particular book has been labeled with so and so number. So the bacteria seems to be doing that for every single bacteriophage or virus that it attacks, so that the next time the same virus attacks it again, what it does is it takes that small piece of DNA which it had already acquired, and hence the immunity part here. It takes that small DNA and it converts into an RNA, and then uses a very, very special protein to make a complex, and goes and attacks that newly the new DNA that comes from that virus, so which is shown on the right side on the top part. So the same virus, once it attacks again and it tries to inject its DNA, the bacteria can now launch a defense, because one, it has a catalog of that DNA, so it knows that it is foreign, and two, it has a protein, which is generally the Cas proteins, which together form a complex, and it chops off the viral RNA into pieces, or viral DNA into pieces. It could be also an RNA, though there are CRISPR RNAs as well. And that's how it fends off this fresh infection. Okay, so keep that in mind, that what we have from bacteria here, and that's what the basis of CRISPR is, a very, very specialized type of adaptive immunity, in which an RNA and a protein, when it is complex together, can be specifically taken to a designed region, or any region of the virus which that RNA is complementary to. So if you have a different region, then it will go and bind to a different region and to chop it into pieces. So now we shift gears and understand what genome editing basically means. So we will come back to CRISPR, because how CRISPR is being used for genome editing is something that we need to understand. So genome editing essentially has 3 steps, which is you recognize a target, in this case the DNA, you add a modification to that target so that you correct it, and then you introduce that DNA, that modification, and your DNA is now changed. And that's what I meant when I gave the term rewriting the code of life, because for a long period of time it was almost assumed that we are a product of our own genes, and there is no way to change that, and we are kind of a little bit playing God over here when we are really changing the genes. So even to simplify this, you can almost consider it in the form of a typewriter, or a MS Word. Typewriter is quite old, so a Word document where you have written something which is your genetic code, and where somewhere there is a kind of an abnormality, and instead of writing the, you have written AHE, and you want to change that A back to T. So essentially what you need to do is you need to take the cursor right there, you press the backspace, delete the A and introduce a T, and then you save the file, and then your file gets saved. So genome editing is almost identical to how you would work on a word processor file. The cursor here is the CRISPR complex. The backspace is of course a DNA break, and then a T has to be added in the place of A, and that T is your what I would come in the next slide as a template which you are adding to the DNA. And because the DNA replicates and new cells are formed, and this DNA goes into the new cell, so it becomes heritable. So it gets into the daughter cells and is saved permanently inside. So it's a completely heritable form of change. But the questions are, of course, where are you doing this? Are you doing this in the somatic cells? Are you doing this in the germline? Are you doing this in the embryo? How are you doing this? Are you going to take the cells out of the body, do the genome editing, put the cells back, or are you going to throw in some kind of a genome editing tool inside the cell and do it? And most importantly, why are you doing this? Are you doing this to correct a disease which you have and you want to increase the lifespan of an individual? Are you going to prevent it? You think that there might be a disease, and therefore you do some correction to make sure that the individual doesn't have a disease. And of course, this leads to more complicated things. Are you trying to improve certain types of traits? Like you want to have a person with, let's say, more muscles, that who is very strong, who can constitute a complete army of more efficient individuals, or let's say change the pain receptor so that a person doesn't feel the pain. All no science fiction, these can actually be done. And most importantly, design, and we all know what the complications associated with design. So just like we are talking about genome engineering, it is also important to talk about all these questions, because these determine whether what type of work in terms of genome editing is really for the benefit of the community, which is not just at the level of the scientists, but also about the global community who is looking up to genome editing to solve questions that one can actually address much more efficiently. So just a very short video to understand what genome editing looks like. So this is the bacteriophage, which enters a viral cell, and it, like I told you, that it has its genetic material, in this case the DNA, which it introduces into the bacterial cell, and this genetic material is now then recognized with a Cas9 complex, which is the protein here, the Cas9 protein, and then this Cas9 protein actually, as you would see in the next slide, it will make a cut. The Cas9, and there is an RNA component to it. It hybridizes, and then it generates that DNA cut, and that's how the viral DNA is cut. As it appears, you can use the same principle also in the case of a human genome editing, where you specifically design that cut to happen on a certain section of the DNA. Okay, so this is the kind of the business end of the story, which is a Cas protein, and it has got an RNA component, like I told you, and this comes from a very early paper. I mentioned Prashant Mali already. This is from his paper in 2013. 2012, of course, was the year when the first paper from Doudna's and Charpentier's group came. So how does this whole thing work? So you have a Cas protein and you have a Cas9 enzyme, which is an endonuclease, and you have this blue RNA thing. And this RNA basically can be directed to go and recognize any double-stranded DNA as you like. So there are 20 bases of that RNA, which is shown in dark blue. And once it goes and specifically kind of binds to that target, the Cas9 then causes a double-stranded break, and here it is shown in the form of those kind of welding machines, and this location of this break is also very precise. Now, once it breaks the DNA into two parts, the cell, of course, doesn't want to have a condition in which its DNA is broken. So what it does, it tries to repair it quickly, and in the process, it can do either a quick and dirty job, where it tries to simply bring together the ends of the chromosomes and repair it. Alternately, if you put in a small piece of DNA which has got significant similarity, or as we call in biology as homology, to that site of the break, then that small piece of DNA can get introduced. So the first process would cause things known as insertions and deletions, which are not good, because you are actually changing the genetic code into something which is uncontrolled. And the second one is actually a very precise regulation of the break to introduce a foreign DNA. Now that is very, very significant if you consider it for therapy, because supposing you have a BRCA1 mutation, you want to correct it, then you could actually produce a small piece of DNA which has the correct version of the BRCA1 mutation and introduce that, and that way you can theoretically correct the mutation inside the cell. It seems very fascinating, but in reality, it is not as straightforward as one thinks, because in order to have all these repair things happening very precisely, you would need to have the donor DNA, and the DNA break which has happened, the CRISPR complex, and the recombination proteins all to be present at the same point of time in the cell. And most importantly, there are 20 bases that I told you about of homology, which the RNA has to the target DNA. As most of you have done probably polymerase chain reaction in the lab, and you use primers to amplify something in the genomic DNA. In addition to the target, we oftentimes have a number of other products, and that happens because what is known as off-targeting. And off-targeting essentially is a process by which even a partial homology to the site can give rise to product. So the guide RNA actually behaves like an off-target, and instead of, let's say, targeting the BRCA1, it goes and cuts the P53, which of course is very, very dangerous. So therefore, there's a lot of research in trying to make this whole process more foolproof and more bona fide in terms of outcomes. So that's the part of research that has been going on extensively, and my lab is also interested in doing this kind of research, in trying to make this process much more efficient, so that genome editing is safer. But of course, the applications of genome editing can be huge, and people have actually made corrections at the level of sperm or in the ovum, and generated transgenic animals which are having different characteristics altogether. People have designed guide RNAs against all the 20,000 genes and the 1 lakh non-coding RNAs that are present in the human genome, and have knocked out each and every gene and tried to see how, let's say, drugs interact with certain protein products, or how to make even better and more efficient drugs. So those kind of studies have been done. You can use a Cas protein which doesn't cut but rather simply binds to the target site. And once it does that, if you add a fluorophore to that Cas, then you can light up certain genetic elements. In this case, you are actually looking at the telomeric loci, which fluoresce and give this kind of a punctate pattern. So Cas9 can be used to illuminate the genome, like you see. And if you're smarter, then you can even make genetic circuits. George Church is a pioneer where you have almost a logic circuit, like a computer logic circuit, put into the cell. So supposing you want to make a cell auto kill itself if it is cancerous, so it recognizes if it has got a mutation which causes cancer, and then the CRISPR can be actually guided through a circuit of gene A and gene B and gene C products, so that a poisonous gene can be produced, or a poisonous protein can be produced, and the cell can auto kill itself. So fascinating work which are done on the field of chemical and synthetic biology, where CRISPR based genetic circuits have also come. I touched upon this already, that the field is so vast. It encompasses almost currently every single field of natural sciences, I would say. I mean, there are CRISPR experiments being done at the International Space Station, and CRISPR experiments being done to bring back the woolly mammoth back to life. So the ability to change the DNA of an organism brings so many different applications that it is almost it's considered as probably the discovery of the decade, in terms of what it can do. That what is it that you cannot do, or what is it that you should not do. And we have also heard about the stories about embryonic genome editing, which happened in China. We only probably know about the tip of the iceberg. There are probably such experiments being done in a lot of different countries, but it raises a lot of ethical issues as well. In this case, for example, the editing that was done was actually to make this next generation resistant to HIV. But it was pointed out that the same genome editing would cause it to be more susceptible to, let's say, West Nile virus infection. And at the level of let's say ethics, in such a case, you do not actually have a choice as an unborn individual to really have the decision-making authority to say whether my gene should be edited or not. So it's kind of touching upon so many different aspects of ethics and ethics in biology and so on, that these kind of discussions are there. At the moment, there's a moratorium on human genome editing across most countries, and there's a kind of an unwritten law that no such experiments would be done beyond the 14th day of the developing embryo, and even to get a let's say the approval for such an experiment takes a lot of effort. So yeah, so that's the thing, that the evolution of CRISPR has happened since 1980s, and we are at 2020, where we are basically curing diseases. We are generating improved crops. We have developed better and better versions of the CRISPR systems, and which is actually helping in so many different formats. And now, of course, CRISPR diagnostics, which I'm going to tell you in the later slides, is also something that is coming up in a big way. Probably the most significant application of CRISPR has been in the level of therapeutic genome editing, and doctors altered a person's gene last year using CRISPR for sickle cell anemia. Sickle cell anemia, of course, is a huge problem all across the world, and the strategy in this case was that it would be the blood would be taken out and edited and then returned to the patient. And there were companies, multiple numbers of them, that have formed. But the good thing is that this was from last year, and this is this year, the first patient who received CRISPR doses, Victoria Gray, is actually, let's say, medically cured of that disease, and there has been no no complexity associated so far. I should point that out here, that it's just a 1 year of follow-up, so it's still early, since it would require many years of follow-up to understand what are the long-term effects. But the symptoms of the disease are not there, the blood transfusions are not required, and it's kind of championing the whole cause of genome editing for sickle cell anemia, in at least for in the case of in the case of such kind of diseases. However, the sickle cell anemia is not a disease of the US only. In fact, if you look at this map, India seems to be even darker red than that of US. And that's because sickle cell anemia, as Dr. Abhijit Chakraborty would probably agree, is that it's one of the most prevalent diseases in India. And some estimates, they say that more than 50% of the carriers of sickle cell anemia are from India. It generally happens in a lot of tribal belts in Maharashtra, Nagpur, Bengal, Tripura, and a lot of other places in India. There is social stigma associated with marriages because it's a autosomal dominant. So if you can actually genetically counsel and try to bring down the prevalence of the disease. But the complexities when we started working on this extend beyond just at the level of the medical diagnosis, but also at the level of the social and the political nature, in terms of what happens when a disease is detected and so on. But needless to say, this is a point mutation which is present in the glutamic, it's in the beta globin chain, which causes a conversion into valine, glutamic acid to valine. The cells, the cells begin to grow from their normal circular shape into a sickle format, and then they kind of tend to aggregate and block the arterial pathways, and then there are episodic pains and different other comorbidities which are associated. So when I started my lab after finishing my postdoc and joining here, this was actually, I was very lucky to be a part of this CSI sickle cell anemia mission. It started in 2017, and then we finished the first phase, and now starting the second phase in 2020. And our idea was actually to try and see if one can develop a proof of concept correction of sickle cell anemia in patient-derived stem cells. It would be very, very proof of concept before taking it into secondary steps. And to cut a long story short, I won't talk too much about it here, but basically we developed an orthogonal Cas protein, which is from Francisella novicida, a different bacteria. The most commonly used Cas protein is actually from Streptococcus pyogenes, and people have engineered it, evolved it, and done a lot of different things to make it more specific. But what we found out was that this particular Cas protein from Francisella novicida is actually highly specific in terms of distinguishing between off targets. So you remember this concept of off-target genome editing that we talked about, one of the bottlenecks in genome editing for therapy. And what we found out that if you can see the lower left gel here, if you take off targets with SP-Cas9, Streptococcus pyogenes Cas9, then these are readily cleaved. But if you take off targets with the same number of mismatches with FN-Cas9, these are not cleaved. So somehow this Cas protein has got some very interesting ability to distinguish between what is a target and an off target, and it simply evicts itself, as seen from these single molecule studies, not single molecule, these actually bulk binding studies, where it doesn't bind. And some part of it is probably located in the structure, but we have very exciting data to show that could probably be the reason. And as a proof of concept, of course, we took sickle cell anemia patients, we converted into their iPSCs, corrected it, and showed that yes, indeed, this can be really done. And we published this last year in PNS, and it was actually highlighted as one of the main articles in that issue. But this was more academic than anything else, because we have a disease to cure over here, and correcting in iPSCs cannot be the way forward for a medical treatment, because iPSCs have other issues in terms of let's say chimerism and possibility of cancer causing mutations to come in. So we really went and thought that if we had to bring this to people and bring the first trials of genome editing in India, what it would need to actually correct the disease. And we talked to clinicians and read literature, and it seems that we need to correct somewhere around 10 to 30% of the stem cells coming from the blood, which is the hematopoietic stem cells, in order sure that a patient doesn't give any chance of disease, or let's say the patient doesn't seem to have any kind of manifestation of disease ever, if you correct them at the stem cells. And that is the percentage that we needed to reach. And how we would do that would be to take out cells from an individual, correct the cells using CRISPR-Cas, and then bring those cells back into the patient. Of course, it seems that it's a very complex thing, because there are so many parts of this very small cartoon here which have to be figured out. For example, how much of hematopoietic stem cells, where to do such trials, how to get the cGMP grade Cas protein and things, and then most importantly, what are the immune reactions in the patient, what are the treatments that have to be done, but most importantly, how you would deliver this Cas protein inside these cells. And I always mention this particular paper which came in 2018 as a very, very how to say, rapid development of an antisense oligonucleotide therapy for a rare genetic disease, as a means to understand that if you have an entire system which is supportive and which is actually moving in the right direction in terms of trying to provide beneficial healthcare, then such kind of trials are also probably not going to be impossible. Of course, it also has its challenges, but in this case, you had a kid who was diagnosed with a rare disease in 2016, and within 2 years, in 2018, when the first dose was given, all the preclinical studies, including the safety, toxicity, and everything else, was actually done. So that gave a lot of hope that yes, indeed, if you can bring together experts in such kind of an activities, then we would probably be good to go. And we realized that if we bring in FN-Cas9 for sickle cell correction in patients, we would of course need a higher efficiency variant, because the wild type Cas9 from bacteria was not good enough to give us those levels, 30% or 10 to 30%. We would also need appropriate vectors for delivery, so that it can be delivered inside the cells, and most importantly, robust preclinical data that would be important to convince the regulators going ahead. And just to cut a long story short, we started making fruitful collaborations with different people who are really the experts in these areas. With Osamu Nureki at University of Tokyo, we started a very fruitful collaboration where we started engineering the Cas protein to increase the efficiency of gene correction and also increasing the flexibility of targeting. He interestingly has solved the crystal structure of Cas9 and is one of the pioneers. With Mark Walters, David Martin, we started working on the in vivo validation. With Anand Swarup at NIH for adeno-associated vectors, and of course, whom I have not mentioned here is Andre Lieber at at Seattle Washington University to develop adenovectors, whereby these things can be made into a vector-driven form. So to again, to just briefly tell you, so we have been able to do all these things. There is an enhanced FN-Cas9 which has now been generated. Currently filing the IP things with that, and it's possible now to have this particular Cas9 to be more efficient than the wild type Cas9. So that also not only gives some scope to go into clinical trials, but also at the level of having some amount of control over what is about the patent landscape as well. So with that, I come to Feluda, which is an extension of that work, and almost all this work is done in collaboration with kind of my mentor here at IGIB, Dr. Shobhik Maiti, who is a nucleic acid biophysicist. He brings all the elements of nucleic acid biophysics and the chemistry of the CRISPR system, and together with the molecular biology that I bring in, we have kind of made a pretty big and formidable team now to address multiple aspects of CRISPR-Cas9 based editing. And that the whole concept of Feluda, which is FN-Cas9 editor linked uniform detection assay, came from discussions about how the Cas protein can specifically recognize a mismatch. Like I showed you that it doesn't cleave the mismatch targets, so therefore it specifically recognizes a match or a mismatch, and then this can be given in the form of an output which is of different kinds. So you have the Cas protein which distinguishes between two DNA pieces which separate at the level of one base pair mismatch. Traditionally, you would not get that amount of specificity with any other thing other than Sanger sequencing. So our idea was to actually develop a CRISPR-based diagnostics which could work in the field and which could still be used for genotyping individuals to bring the power of genotyping at the hands of the individual at a very POC level, so that things such as carrier screening and actually genotyping diseases can be done. And that's where we went in with FNCAS9 because it has such a high specificity of matches and mismatches. And we thought about, had adapting the output to various forms, like you see over here, either in the form of a gel or a capillary electrophoresis or a fluorescence readout, or as you will see later for COVID-19, we used a lateral flow. And the principle is that, like I said, you have the sickle cell anemia, which is a GTG in the place of GAG. If you introduce another synthetic mismatch in the guide RNA and you walk the entire length of the guide RNA, you would find very specific positions in which you would have a completely diametrically opposite readout for a wild type gene versus a sickle cell substrate. And a sickle cell substrate would be cleaved, whereas a wild type will not be cleaved. And this would be our readout. And that readout is very robust. It works for every single mutation because the properties of distinguishing is not at the level of the sequence, but at the level of the Cas protein itself. So you simply have to design another guide RNA against another gene, and you can target essentially all the 70,000 or more Mendelian disorders that you can potentially think of. And like I said, that it really can detect all DNA RNA variations, including zygosity. So you can, we did these blinded studies with patient samples coming from Raipur, where it was able to distinguish between what was a sickle cell trait versus what was a sickle cell anemia and what was wild type with 100% accuracy in blinded samples. You can convert this into an affinity-based PHENUDA, where instead of the agarose gel, you can have in the form of a detector which detects fluorescence of Cas9 binding. We combined fluorescence chemistry. And it's very, very important to see what are the applications of it. We started working with Govind Makharia, Dr. Govind Makharia at AIIMS, who came and told us one day that we have a lot of people coming into the OPD with complaining of Helicobacter pylori infection, and then we give them rifampicin, which is the first line antibiotic, and several of them actually the bacterial cells have got a mutation which make them antibiotic resistance, and these antibiotics do not work on such patients, and they complain even more. And normally what you have to do is you have to take a biopsy of the stomach, take out the bacteria, culture them, grow them for 3 days on a rifampicin plate, and then understand whether they are resistant or not. We could design a PHENUDA assay where this can actually potentially be done at the OPD within 45 minutes or so, where you simply take the Cas protein, take the bacteria, take out the DNA, and do this. And we really wanted to make this into a technique which could be done at levels where normally sequencing would be unheard of, or at least sequencing results would be too costly. And to do this was something that was Dr. Shobhik Maithi's kind of real idea behind developing PHENUDA, which is to have it as a POC CRISPR diagnostics in the field. And normally, what the current methods of doing a very crude test of sickle cell anemia is a solubility test, a coagulation test, which is not a confirmatory test. And you take blood, and then you have this kind of a turbidity or not, which you see. And this was in Raipur in a sickle cell camp in a tertiary healthcare center, where children are lined up here to actually give blood. If you look at this kid who is looking very curiously from the center of the queue, she doesn't have shoes on, and she doesn't seem to be too thrilled at the concept of really getting her finger pricked and giving blood. So we came up with an idea that you can ask this kid simply spit it to a small tube, and classic Maniatis and Leninger textbook, you can put in buffers that lies open the cell, get the DNA out, perform the PHENUDA within half an hour, 45 minutes, and whether you have a trait or a disease very quickly using the CRISPR diagnostics platform. So this was where we were at the beginning of this year, and we were thinking that we can simply put this all together and write up a paper. And that was what we initially intended to be. That we know that how difficult it is to get technologies out there, the complex routes and so on. But then, of course, COVID struck, and life turned upside down, and we realized that the entire assay can be even made better, or let's say made more suitable for COVID-19 diagnosis, simply because it works on the principle of a Cas protein binding to DNA. In this case, it was an RNA virus, so you needed to convert it into DNA. So that's how the Feluda was designed, where you would have the swab or the saliva sample taken, specimen collected, and RNA extracted. And this RNA extracted would then be converted into a single step into DNA, and a PCR reaction would be carried out. Now, this is called an RT-PCR, not to be confused with real-time PCR, but a reverse transcription PCR, where the DNA is the RNA is converted into DNA, and then the amplification is done using very, very specific primers which are biotin labeled, and they generate products which are biotin labeled, and then we combine the CRISPR biology with the paper strip chemistry to give rise to a visual readout on a band, where there would be a control line on the top which tells if the strip works fine, and there will be a test band at the bottom which tells if the patient is positive or not. So it was rapid because you can do all these things within 45 to 50 minutes. It's economical at the lab level. It doesn't cost more than 300 to 400 rupees for the reagents per se, and it doesn't require a specialized equipment like an RT-PCR machine, but can be done using any PCR machine which is present in pathology labs, universities, institutes, and even on as I will show you in the next slides, in PCR-free versions of isothermal PC amplification. So just to go, this is how the commercial strip looks like, where you have the complex of Cas9 tagged with tracer RNA, and then it recognizes a biotin substrate if the sample contains the COVID-19 DNA, and then it moves out, and then there is a test band here which has streptavidin in it, and the biotin and the streptavidin interact, and then they give rise to a band, and this is how it looks like. Finally, you would have a positive sample containing both the bands, and a negative sample not having any of them. And it's very interesting that this year also has seen the passing away of our favorite Feluda character in the form of Soumitra Chatterjee. And as a Feluda enthusiast, it would not be complete without having his sidekicks, which would be in the form of Topshe and Jatayu. So Topshe is, of course, an app that we made in collaboration with Aduvo in Chennai, where you could simply take a picture of the strip and you get the readout. It's very, very sensitive, so it can go to a few copies of the viral substrate. And of course, Jatayu is an artificial intelligence-based diagnosis where it's junction for the analysis of your Feluda assay. That's the full form, where you can simply put in your sequence of interest for whatever disease that you want, and a Feluda primer and the Feluda design will be made. I told you already about the fact that you do not really need to have a PCR machine. This is not one which is coming to the market, of course. The market version currently requires the PCR machine, but there is also the prototypes for the second and the third versions, where we do not need a PCR machine, but a simple heating block and the combination of isothermal amplification in a single pot would give rise to COVID-19 readouts very, very quickly within half an hour. And these are just some of the costs for the reagents that you see over here. I would not have to go into details about, of course, it has got a lot of media attention, probably much, much more than it should have, because, similar kind of innovations are happening all across the country and across the world. But we have learned a few lessons during this whole process. One of the good things, of course, is that, DCGI ICMR has approved it to be a replacement of the RT-PCR. So that tells you how sensitive it is in terms of detecting what is a positive or a negative sample. But we also learned a little bit about product development. How do we go from an idea to a product? And this has been the timeline, really. In January 28th, we sat together, and I'll show you there's proof for that we thought that Feluda could be adapted for COVID. And by September, there was a DCGI approval for a new company that was formed in which is Tata Medical and Diagnostics for the market release. And I know people are very impatient and all, but normally such kind of things probably take a few years, as I am told in certain cases. That of course doesn't tell much about, or is not to say that we have worked extra hard, but it also tells a little bit about how things can be pushed. In the case of a pandemic, it kind of gives a kind of impetus to work even harder than what we would probably normally do. So this was January 28th, and we actually started working on this before the first case of coronavirus that was reported in the country. We formed a WhatsApp group, and we decided to make Feluda for coronavirus. And then in March, we had this ready in terms of showing it in synthetic DNA. We got access to the first samples actually that came in Delhi from NCDC, and we started looking at them and seeing whether this whole concept works. You see the control band developing and the test band coming up at the bottom here somewhere. And of course, in September, this was approved for use in India. Now, one of the lessons that we learned, and I just add this because I think that brings in an element of humor also, is that, as scientists, we often speak a different language altogether between what the corporates speak, in this case, companies. And, they have different types of deadlines, they have different types of requirements. And sometimes we are unable to cross over the hurdle of translation because we do not have a good Google Translate in between. Fortunately, in case of Feluda, we of course had Dr. Anurag Agrawal, the director of our institute, who would play this role very efficiently. And we learned a lot of different things during the way, such as, what is the quality of the reagents being equivalent to how the final product would read. And, things working in the lab does not necessarily mean that they will work every single time in the hands of a consumer. So you have to go through all these processes of quality control, which are sometimes not in your hands. Supposing you come up with a primer batch that is faulty, and there is no way to know where the problem actually lies. And another thing that we learned is that when we are measuring the accuracy of a test like this, and you're using the quantitative real-time PCR as a gold standard, which itself doesn't, at least we have now evidence to say it probably sometimes doesn't even qualify for the bronze standard, then there are issues in what you consider as an accuracy in terms of sensitivity, specificity. So a lot of learning curves, a lot of things to ponder about, a lot of ideas about how we need to think about going ahead for similar kind of preparedness for other diseases, is what was the take home message from the Feluda story. The other take home message was that, definitely our students are our biggest asset. It's very important that we take care of them in different ways, whether it is releasing fellowships on time, to understanding, how to keep them working in an ecosystem inside India. Because these are the students who actually decided to stay back during the entire pandemic, put in all their efforts to get a product format of a science out. And, they're still doing that. I mean, there is a lot of work still going on in terms of innovations. So definitely, if we have to probably do a science which is relevant and which is useful, we need to have warriors. And these are the type of people who would, and I can almost tell you that most of them have been COVID positive during this whole process. You know, they say that raising a child requires a village, but if you have to bring a product to market from the lab, I think you require the whole organization, because every single person's role here counts, whether it is the logistics delivery boy to bring in the courier from the airport to the lab, to the highest officer in the form of the director general or the director. Everybody has to work in sync, and this was something that happened, at least in the case of Feluda. So really thankful to a lot of people who really put in all their efforts. And partly because probably the name, it from the day one has got a lot of attention. I wished it did not, but social media has been one of the major areas where there has been a lot of attention. Particularly if people take notice of your work, we need to probably begin to ignore a lot of people around there who keep on asking things in a negative light. But again, social media can also make science accessible to public like no one else. And heartily, on the day that it got the approval, we found that Feluda was trending after Sri Krishna and press freedom. I mean, look at science. That's probably one of the very, very let's say red letter days as far as science in Twitter in India is concerned, which was fascinating to see. And when the Shashi Tharoor comments on something like this without using a single big bombastic English word, that tells you that probably science is going in the right direction. But of course, like I said, that these are nothing compared to the actual work, which is to take it to the people, to help them. And pandemic, of course, is the mother of invention. I'll just show you that you have to keep on improvising, keep on innovating, because the solutions, just like there is not a single vaccine developed, the tests will also have to evolve to make into something where it can actually go at the level of the home or the office, because you still have to give your samples to the lab. So how would Feluda maybe in the future look? It would probably look a bit like this, where you would these pipetting steps would be completely eliminated. They would come as pre-pipetted tubes. But the major drawback that we need an equipment, which is the PCR, and how to do it at home. You simply put it inside your elbow or inside your palm, and then you use the body temperature. We have this prototype in a manuscript and public domain, so you can have a look. And this for 10 to 15 minutes will make sure that the PCR has happened. And then you simply put in your lateral flow buffers and the strip into it, and then get a readout after some time how it looks like. So all these innovations will keep on happening as it is happening. We are working to develop into formats which are much more point of care, much more easily applicable. And that's the last slide, probably the most important slide. A lot of people to thank here. We have a big group and a lot of collaborators and generously funded. Tata Medical and Diagnostics have been wonderful to really push this into the field as one of the first approved CRISPR tests across the world, actually. So that's a big feat on their part. And they have been supporting also in different ways in trying to make this test even better. So yes, I think with that, I'd like to stop sharing my screen. And if you have questions, please feel free to ask.
Thank you very much, Dr. Debojyoti Chakraborty. Now, I hope you'll be happy to answer questions. So there are audiences from, they are in the YouTube live as well as on WebEx. So we are going to put up, I'll just examine. They are putting on chat mode. So I'll put up those questions to you if any question has come. Just going to that.
Thank you, Dr. Chakraborty, for a very, very interesting lecture, and which of something we could make out, even though we were not really specialists in the field. It's obviously very exciting subject, and I would like to congratulate you and your team for making such an impactful, work, and I'm sure it's going to make a great difference to the country.
Thank you very much.
Before Dr. Maiti puts the experts' questions, as a non-expert, I want to ask you one or two questions.
Yes.
You mentioned two things. One is about the Feluda and its application to detection of COVID. This is an application.
Right.
And you also talked about sickle cell anemia and how it can be addressed using the CRISPR to some extent.
Right.
Now, I want to know whether you or people are working on other diseases caused through genetic issues, including, for example, diabetes and other things.
Right.
Or is the question wrong?
So if you're talking about the detecting the genetic components of such diseases, which is, let's say, mutations that are associated with such kind of diseases, yes, it is. The portfolio is completely infinite here, because you can just simply change your guide RNA and look for a new disease. As a single lab, our interests are more in developing the therapies. So therefore, we are not actively into taking one disease at a time and then generating Feluda. I think that will now in the future probably work in a company mode. We are in talks with a number of such kind of companies who are interested in bringing these as products. So, but yes, the biology part is of course done. So we know essentially what are the principles of design and development. So it totally depends upon what is the final aim. If somebody is interested in bringing this for diabetes, let's say, definitely such kind of panels can be designed which are specific for diabetes.
Okay, second question is about the way you can detect at home, which you mentioned at the end. That is, I feel, that of greatest interest to the entire audience, including some of us. So you were a little fast at that time. Can you just repeat what exactly you mean by you can do the test at home? What are the
So
elements
I
of the
think
test?
it's something that it's not something that we came up from ourselves. It's well known in the field that there is a alternate version of PCR which is called isothermal amplification. There are ways by which you can do a PCR without a machine. You can do it on a heating block. You can do it on a water bath, which maintains at the same temperature. And we are using the same principles over here. We found out that the human body itself can generate temperatures which are very ideal for doing a PCR at a single temperature. Question is whether that is can be done at the level of an individual who has no knowledge of biology or chemistry in let's say an Indian village. Hence, it's although it is practically doable, but to really make sure that it can be done at home in the correct fashion would require a lot of iterations in different levels. So we are working around it. I mean, whether it would be a kind of a model where somebody comes to the house and does it, or whether it would be doable by the person, because not everybody probably would be able to appreciate and understand the entire technology unless it is almost as simple as a pregnancy test. Which currently it is not, because a pregnancy test detects antibodies. Feluda requires a PCR. So therefore, how to get rid of those complexities is what we are working on.
Thank you. Now over to Dr. Maiti.
Yeah. Mostly I find they say many people have just told that it is an excellent talk and congratulations to the speaker and his team. But one question somebody has put it from IIT Palakkad. Yeah, he says that whatever experimental work you have done, the changing and all those things, whether one can simulate. Is there any simulation procedure available to that or not? That is one part. I think that there may be a student who has asked this question from IIT Palakkad.
So I'm not exactly sure what is meant by simulation over here. If you are talking about of simulation at the level of whether protein will bind or not bind, yes, those kind of simulations can be done, and it's available already. Many people have done these simulations, but I'm not sure if he or she means the same thing or not.
Okay. Okay, thank you. And
May
one
I
more
ask
question has come.
question, Philip?
Okay, please add.
Yeah, thank you, Dr. Chakraborty, for an excellent talk. This is B.S. Tomar. Again, a layman's question.
Yes.
I just wanted to know whether the same methodology that CRISPR-based methodology can be used for development of a vaccine.
Well, not the direct way, because the principle of vaccine is that you develop something which can give rise to an immune response. CRISPR can play a part in it. So, for example, CRISPR can make sure you can find out, let's say, which are the portions of the viral protein which gives rise to more rapid immune response by knocking them out, changing their sequence, and so on. So, CRISPR can aid the process. But a CRISPR vaccine, I don't think so. I don't know how that would work. Although a CRISPR drug would be something that one might think of, where you can develop a CRISPR complex that goes and specifically chops off the SARS-CoV-2 RNA. Such kind of activities can definitely be tried out.
Thank you.
Okay, I'll add now one Saili Mule. She has put it from a data analysis background. I would like to know how can data analysis be helpful in terms of getting more information out of genetic domain and going from science to production. She's from data analysis.
I think the entire work is all about data analysis, really, because data is the person who has data is probably the most powerful person at the moment in the world. So all the sequences that are being deposited, whether it is viral sequences or whether it is humans affected with the virus, they all come in order to understand how your probes for detection are going to be, whether these are getting mutated, whether they can be changed, and so on. So I think those are the aspects which are continuously evolving. And more data are coming in and making the whole process more fruitful. So yeah, I think data analysis plays a very, very important role.
Okay. Another question from Maishuru, RMP
Yes.
Maishuru, Dr. Sok, he has put it this way. What is the difference in specificity and sensitivity in comparison to RT and PCR testing?
Sorry. Yeah. So specificity and the sensitivity of the Feluda is at par with the RT-PCR. So I think when they did the third-party evaluation at DCGI, they got something like 96% and 98% sensitivity specificity respectively. Of course, the sensitivity and specificity is a relative number, so it will vary within that 95 to 100%. In our hands, we often have even 100% sensitivity as well. So it is at par with the RT-PCR, which is a qRT-PCR. Of course, it is much higher than an antigen test because antigen tests detect the proteins and are not sensitive enough to detect low quantities of protein. So it's definitely higher than that of the antigen test. Yeah, I think it's at par with the qRT-PCR, and that's why the advisory also talks about not requirement of doing an RT-PCR if you are considered as a positive or a negative on Feluda.
He has put another question. Is there a way to detect directly RNA without PCR? Can we use RNA targeting Cas type proteins?
It's been already done. So there has been work from the work of Jennifer Doudna. There is a protein called Cas13 which actually recognizes RNA. The problem actually is at the level of the sensitivity. The reason is that when we do PCR, we're actually amplifying a very tiny signal. So even if a patient with a high CT value who has a very few copies of the viral RNA, the PCR makes sure that there are many copies of that RNA when converted into a DNA is made, and therefore you are getting a signal out in the final phase. If you try to detect it at the level of RNA, there is no technique where you can do an RNA amplification. Well, there are one which is a bit complicated, but you have to basically find a way to amplify the signal. You can amplify the signal either at the level of detection or you can amplify it at the source. If you try to amplify the detection at the level of the detection, then you are also going to increase the noise. So all these complexities make RNA direct detection not so sensitive. But all said and done, I think there are people who are trying to work on that, including us. Let's see what comes out of it.
Okay. One more question has come. Are you all working towards detection of any other viral diseases like hantavirus or many others? Will there be any difference in concept?
The concept remains the same every single time. We just have to change the guide RNA sequence. So the concept doesn't change much. So if you ask me whether we are working right now, no. But like I said, that this in the future it will probably go through a different route, not necessarily academic, in trying to bring out panels of Feluda, depending upon how much acceptance it has in terms of the people whom it will serve. That would be my answer, but not at the moment. No, at the lab.
Okay, thank you. I don't find any other question. I just ask Dr. Abhijit Chakraborty, do you have any question to put on to Debojyoti?
Saibal Basu wants to ask.
Achha, okay.
I have a question. Yeah, I can understand. I'm Shaival Basu from HBNI. This is the So this is regarding the Feluda is, I understand, more of a very accurate detection system. How far is it from gene editing? I mean, how it can aid the gene editing? Because that's something which is in a future possibly a much larger implication. But I am completely zero in this field, so I would like your opinion on that.
So, like the first part of my talk said, that we are still committed to using FNCas9 for genome editing. That is the goal. That is the real thing that we are committed to working on in the next few years. Start with sickle cell anemia and then going with the other diseases. FNCas9 has got some very, very interesting properties which we want to exploit, mainly in the terms of specificity. And we will see how this goes, because bringing it into a form of a clinical trial for a disease has to go through a number of different hurdles, which are regulatory. So we will try to see how far we can take that. We have, there is a lot of promise in the initial stages.
You can ask Abhijit.
Okay, Professor Abhijit Chakraborty, do you have any question to put on?
I don't have a question. I would like to really congratulate you for this excellent work.
Okay.
Thank you.
Thank you. Okay, in between, I have got one more question from Pratibha Gupta. Can it help patients of Alzheimer, Parkinson's, or such patients? This
So, like I said, that if it's a genetic disease that you want to detect or you want to correct, so there is a mutation in DNA or RNA, then Cas proteins can specifically go and let's say latch onto that, and you can then have an output, whether in the form of a corrected cell or whether it is whether in the form of diagnostics. Problem is, the disease itself and the complexity associated with it makes a lot of things still at the level of only research and development. Alzheimer's is one of them, because even if you could correct a mutation that causes Alzheimer's, it is a late onset disease. The mutation is always present in the person from childhood, but the onset is of course late. And if you even discovered a way by which you could do that, how would you deliver it into the brain? There are a lot of hurdles there. But science is progressing very rapidly, including at this in this field. So who knows? I mean, there would be something coming up in the next few years, maybe.
okay. So there are no more questions. Then I
There's a
thank
question
Dr.
where what are the false positives encountered with Feluda? So the false positives generally happen if there are two reasons why false positivity happens. One is that if the Cas protein is recognizing something else, which is let's say not the SARS-CoV-2, but still recognizing and giving a signal. Or two, if there is a chemical false positive where the line, which is the control, the test line, actually develops. So to answer the first question, we did this with at least 10 different types of very close bacterial sequences, which not vector, viral sequences, which causes other diseases like influenza, H1N1, and I can some of them are also in the paper. So this Cas protein is highly specific to a point mismatch. So if your 20 bases is not matching by even a single base, then the Cas protein doesn't give a signal. So that's an advantage of using this kind of an assay. And if it's at the level of the chemistry, where you would have some kind of a non-specific binding, I mean, maybe once in a while in a batch of not so good chemicals, one can see such kind of a faint line developing. But this is as prevalent as you would have a false positive that you would get in any biological system. So it's entirely a matter of retesting or testing it using a different batch is what I would guess.
Okay, in between, another interesting question has come. How to dispose the test kit at home?
We don't have a test kit at home yet, so that answer I think I won't be able to give now. But I mean, if there is a solution for home, it would be an end-to-end solution, which would also take care of this aspect. So we will see. Yeah. The challenge right now is to actually, to be very honest, if I can tell you, the challenge right now is to make sure that there is something which every single individual, regardless of their let's say their intellectual ability, can simply follow a protocol and do. And that's a huge challenge. Really, we kind of underestimate it because we are all scientists here. But if you have to make something which is very, very reproducible, works in the hands of every individual, it's a challenge.
So maybe I ask a question, and that is, how stable are those strips in, and what are the storage conditions of these strips?
So the strips are very stable. They are stored at room temperature actually. They remain at room temperature, and the stability of the protein varies anywhere between. So it is quite stable between 10 degrees to 44 degrees. And you can generally, the kits are now being shipped in ultra low storage temperatures. I think -20 is probably the
Paper strips or something else?
the strips are being stored, they are shipped at room temperature. The rest of the components, the enzymes and the primers.
Okay, is there any other question to put up? No, I don't find any other questions.
I'm not
So
sure what somebody meant mini PCR here. Is it some kind of a PCR machine or?
Huh? Okay.
It works with
So
every
we
PCR
saw
machine. Yeah, it doesn't. I mean, there is no reason why it should not work with a different format of a PCR machine. Yeah.
getting somewhere.
Post-COVID
Okay.
complications, not yet. Just breaking our head to solve COVID here. So not yet. We have not yet thought about it.
Acha. No, both I am looking at. PCR machine. Yeah. Meaning there were some questions. Okay. So if there are no questions, we firstly take our hearty congratulations from HBNI staff. It's really wonderful to know about your work and such a quick span of time, for short span of time, you have developed a product which will be useful to the society. And thank you very much for accepting our invitation and giving this talk. Thank you.
Thank you very much. It's an honor. Thank you very much. Bye. Bye.